National Mental Health Awareness Month, National Creative Beginnings Month, and National Cancer Research Month. And to celebrate them, I made this brief presentation about mental health research, neuroscience, oncologic research, and creativity from my experience. And this will be like a supplement to my May edition of the LinkedIn newsletter, The Biomedical Artist, because science is art and scientists are artists. They apply creativity to solve different problems, and they take photographs, which are a form of art. They make figures, and so research is in essence art if you apply your creativity. And so where to start? That often I'm asked that I have done research in different fields such as immunology, oncology, neuroscience, psychiatry. How do I make this transition? And I want to say that they are all connected, and my path is very logical. And it led me to believe that the same biological pathways in molecules are important for different systems, and important in immunology, and in oncology, and in neuroscience, and psychiatry. And so, when you have done research in several fields, you see the big picture, and you make a connection, and this is very useful in drug development, because a drug acting on one system, as is known, could act on other systems. It could produce specific effects, and it could produce undesirable and side effects. And so, to start with the example of Calmodulin. Calmodulin, as is known, is the main calcium-binding protein in eukaryotic cells. It is highly conserved, and in humans it is encoded by 3 genes, Calmodulin genes 1, 2, and 3. And it is involved in diverse processes such as cell proliferation, cell differentiation, the action of different enzymes. It is involved in the release and action of neurotransmitters, and in autophagy of cells, and many other processes. There is a review I will provide at the end of this presentation, several papers that you can refer to. This video presentation is intended to be not an exhaustive scientific presentation, but just to summarize my experience on this topic, and to celebrate Mental Health Month, Oncology Research Month, and Creative Beginnings Month. And so, when I was working at Wistar Institute, I was constructing genetically engineered via PCR and baculovirus expression monoclonal antibodies to tumor-associated antigens. They were the EGF receptor and some colorectal cancer antigens. And I was becoming very experienced in molecular cloning, which I also gained a lot of experience at the Karolinska Institute. And all of a sudden, Dr. Weiss from the Medical College of Pennsylvania, Drexel University, called me unexpectedly and asked me if I want to work for him. And knowing that he's a friend of my father, and I'm in a country where I don't know many people, and knowing that I'm not very good at obtaining grants on my own, I welcomed this proposition to work for him very warmly. And when I started to work for Dr. Weiss, he assigned to me 2 projects. The main projects in his lab, one was to study the role of Calmodulin in brain, and the other one was to study dopamine receptors in brain. And as is known, Calmodulin is also involved in dopamine neurotransmission. This is something that my dad has worked when he was a young scientist. And Dr. Weiss wanted me to use antisense RNA for me to construct antisense RNA in order to study the role of Calmodulin and dopamine receptors. His lab already had made advances with antisense oligonucleotides, but as is known, the oligonucleotides have a very short action, and so they have to be administered very frequently, and in the brain this is not desirable. For example, you have to do multiple injections, and his idea was that if I construct a vector that expresses antisense RNA continuously inside the cell after a single injection, hopefully it will have a prolonged effect. And this perhaps in the future could be used for gene therapy. And I was working in cancer research, but I applied my knowledge of molecular cloning to design these antisense RNAs and gradually became introduced to neuroscience, neuropharmacology, and brain research. And since I'm today talking about Calmodulin, I designed Calmodulin antisense RNA and sense RNA by cloning them in metallocyanin promoter inducible vectors, which I obtained from colleagues from the Wistar Institute, Dr. Frank Rauscher lab. And I obtained the Calmodulin genes from the scientist who had cloned them in Japan by writing to him, Dr. Hiroshi Nojima, and he generously donated the vectors to us. And so I constructed an antisense RNA expression vector and Calmodulin sense RNA expression vector from Calmodulin gene 1. And when I transfected PC12 cells with the antisense RNA vector, PC12 cells are rat cell chromocytoma cells which are actually transformed, and they proliferate indefinitely in culture. As the cells stopped proliferating, they formed fewer colonies, and they started extending long neurites, which is a sign of differentiation, which is something similar to what they do if we add nerve growth factor as a differentiation agent to the cells, which was amazing, and it is expected because calmodulin is the calcium binding protein that regulates cell division, and if we reduce its levels, the cells stop dividing and they start differentiating. And so this was amazing, whereas the cells transfected with the sense RNA, they were not responsive to nerve growth factor. They couldn't differentiate. And this became very interesting to Dr. Bill Hyde from the Cancer Institute of New Jersey, a former PhD student of Dr. Weiss, because he is a cancer researcher and physician and wanted to see if calmodulin antisense RNA will inhibit the growth of rat C6 glioblastoma cells. And so I donated my vectors, calmodulin antisense and sense RNA to Dr. Hyde's group, Dr. Chris Brosco and Tony Palmer, and I trained them on the transfection that I used with lipid-mediated transfection, and they found that if they transfect C6 glioblastoma cells with the antisense RNA, the stably transfected clones are less, and they don't divide as much as the normal glioblastoma cells, which they concluded that the calmodulin antisense RNA inhibits the cell proliferation and the viability, which shows that there is a potential to treat tumors such as glioblastomas with calmodulin antisense RNA. So this is a connection between oncology and neuroscience and brain research. And it's the next step because calmodulin regulates many enzymes. In the lab of Dr. Hyde had discovered one of the enzymes that is regulated by calmodulin, calcium calmodulin dependent kinase 3, which regulates eukaryotic elongation factor 2. In a way, it phosphorylates it, and this stops the elongation of the polypeptide chain. But interestingly, it stops the translation of most of the proteins, the abundant proteins. But those that have a less abundant mRNA are translated preferentially because normally the larger mRNAs use the necessary components for translation, and they compete with the low abundance mRNAs. And so I learned about this CF2 kinase, and when I became a fellow in neuroscience at Albert Einstein College of Medicine, I studied the trafficking of AMPA receptors in hippocampus during mQR and NMDA receptor LTD. And to study the differences, my suggestion was to study eukaryotic elongation factor 2 kinase. Why? Because my studies found that both activation of NMDA receptors and metabotropic glutamate receptors induced synthesis of MAP1B, and I wanted to see how we can study the differences in the mechanisms between the internalization of AMPA receptors between these two conditions. And so my studies found that when we block eukaryotic elongation factor 2 kinase, we block the synthesis of proteins including MAP1B, both after activation of NMDA receptors or metabotropic glutamate receptors, but this has significance only for the trafficking of AMPA receptors during mGluR-induced activation. There is more details, I don't want to go into them, but this shows that my studies from oncology sparked my interest in neuroscience. They taught me about the role of Calmodulin in brain, and I became interested to study the role of Calmodulin and its associated enzymes in AMPA receptor trafficking in brain. And this makes sense because AMPA receptors, besides in learning and memory, they are important for the proliferation of glioblastomas, as studies show. The glioblastoma cells secrete glutamate, and this is one of the mechanisms. And also antipsychotics, which are calcium antagonists and block Calmodulin, have been used for the treatment of glioblastoma and other cancers which express Calmodulin. But these drugs are quite non-selective and they have toxic side effects. So if we could design a more selective drug, such as eukaryotic elongation factor 2 kinase, siRNA or antisense RNA, we might block the proliferation of cancer cells in brain or in other tumors, such as breast cancer more specifically. So I don't want to make my presentation very long. I just want to say that having done research in different areas such as oncology and neuroscience and immunology, I see many common pathways, and I think this is an advantage because you can look for pathways that you have learned in a different specialty. Thank you so much for your time and attention. I will provide some references regarding what I discussed at the end of the presentation.